Okay, so again, dopamine is, synthesized here at the terminals based upon the expression of the synthesizing enzymes that work their way down here. Grab onto tyrosine, convert it to L-dopa. The next synthesizing enzyme will grab onto L-dopa, convert it to dopamine. Then the dopamine is going to be packed inside vesicles here at the axon terminal, via the monoamine vesicular transporter, which again would be happy to bring in norepinephrine or serotonin if it were in a noradrenergic or serotonergic cell, but this is a dopaminergic neuron, right? So it's going to pack the vesicles here with the available dopamine that was just synthesized. Dopamine will be released into the synaptic cleft upon the arrival of an action potential. It may very well act at autoreceptors here, actually, that are expressed and usually are linked to some kind of inhibitory mechanism to, hyperpolarize and reduce further release of dopamine. So it's a sort of a way to regulate, the maximum amount of dopamine that can be released basically at this synapse. And then postsynaptically, right? Let's say on a dendrite, you're going to encounter a whole host of potential dopamine receptor targets. And all dopamine receptors are actually G protein coupled receptors or metabotropic receptors, right? And they exert their action via engagement with a second messenger that'll be found inside the let's say the dendritic spine, right, of the next cell. That's called cyclic AMP, and which is actually a chemical that's derived from ATP. So there are two classes of dopaminergic receptor or dopamine receptor. There are what we call the D1-like receptors, and this is there's a different kinds of dopaminergic receptors, D1, D2, D3, D4, D5, but D1 and D5 have a similar kind of mechanism because they're both coupled to a very specific G protein, one called GS. And when the dopamine, let's say this is the metabotropic dopaminergic D1-like receptor, right? So dopamine, let's say, binds here, and then this G protein on the inside, this GS, gets released, and it works its way over, and it can activate this enzyme that's called adenylate cyclase, and it can generate cyclic AMP. So the D1 receptor activation by dopamine will, and that's D1 or D5, or both D1-like, will result in the release of GS. It's a specific kind of G protein, which will then upregulate or increase the availability of cyclic AMP. And this can have a variety of effects, actually. But then there's another class or category of D1 receptor target, again, metabotropic, G protein coupled. But these are what they call the D2-like dopaminergic receptors, and they include D2, D3, and D4. And they're coupled to different G proteins, so they're going to do different things. And the G proteins they're coupled to include GI and GO, but those particular G proteins, when released, by the binding of dopamine, are going to go off and inhibit adenylate cyclase. So they're going to inhibit the enzyme that would generate and upregulate more cyclic AMP. So the effect of the D2 receptor activations is, a decrease in that second messenger. So you've got like kind of a push-pull. You can, through the D1-like, postsynaptic, dopaminergic, metabotropic receptors, you can release a G protein that will result in more second messenger, more of that cyclic AMP, versus the D2-like receptors, D2, D3, and D4. You're going to release different G proteins in the intracellular space, in the postsynaptic cell, that will, result in a decrease in that second messenger known as cyclic AMP. And that has differential sorts of effects on what might happen in these postsynaptic cells.